All right, so smooth muscle. All right, we first were exposed to smooth muscle when we were learning about the different tissues in the body. As you know, muscles are one of the 4 tissue types in our body, and smooth muscle is one of the 3 types of muscles. Obviously, it's called smooth muscle because there are no striations. All right, so this will not have, all right, that traditional actin-myosin sarcomere arrangement that we saw, all right, in skeletal muscle and what we saw in our cardiac muscle. Okay, so smooth muscle is an involuntary muscle, similar to cardiac. Okay, you do not have voluntary control over it. It is operated by the autonomic nervous system. Right? So you're going to see it in a variety of different tissues and organs, like the digestive system. We're going to see it all throughout the digestive system, like the esophagus, the stomach, small and large intestine, all throughout that. The vascular system, okay? It's what allows the blood vessels to vasoconstrict or vasodilate. The respiratory system, we'll see it there. For the bronchioles, same thing. It allows the bronchioles to become bigger and smaller through bronchoconstriction and bronchodilation. So you can see just a couple other places, urinary system, all right? So when we talk about the ureters, which we already know where those structures are, all right, those tube-like structures that lead from the kidney down to the urinary bladder. So they'll help facilitate the movement of urine down to the bladder from the kidneys. And then also in the female reproductive system throughout. Okay, we'll see it mainly in the uterus. Okay, so we're going to talk about some of the aspects. And here's a nice picture that kind of shows you, all right, the locations in the body where we're going to see the different type of places for smooth muscle. Okay, so just to review about smooth muscle cells, they have a fusiform shape, or what we call a football shape, similar to what a fibroblast has, okay? But it's kind of like twisted at either end, so it's going to kind of look like this, all right? Something like that, okay? So it's going to be thick in the center here, all right? That's where our nucleus is going to be, all right? And it's similar, all right, to skeletal muscle, in which we call the plasma membrane a sarcolemma. But in this case, the sarcolemma for smooth muscles have these special types of calcium channels here, all right? It's both voltage gated and chemically gated, all right? But check it out, important aspect. There are no transverse tubules, okay? No T-tubules are present. But we have another structure, all right, which is somewhat similar. We call those calveolae, all right? So instead of like the T-tubules, like a well that goes directly down, all right, the calveoli is going to be like this flask-like structure here, all right? Probably not doing it justice by drawing it in there, okay? And we'll talk about its purpose here, all right? Very little sarcoplasmic reticulum, all right? Remember, sarcoplasmic reticulum is where we keep our calcium, all right, when we're not actively engaging in a muscle contraction, all right? So a lot of the calcium that the smooth muscle is reliant on is going to be outside the cell, all right? It's going to be the interstitial fluid, the fluid that surrounds the cell there, all right? So as we take a close peek here, all right, at the smooth muscle, right. Here you can see how, all right, we have these fusiform shapes, all right, nice and big in the center here. All right, a couple of the structures that you're going to be looking at here, all right, when we're dealing with our smooth muscle cell, as you can see here, the calveoli, all right, those are those indentations that you're seeing here, okay? And then on the inside here, all right, we're going to have several mitochondria, obviously, for the production purposes of the ATP. All right, but we have these structures here, all right, if you look closely, all right, this triangular-like configuration. And this is going to be kind of what our contractile unit is going to be made up of. We'll have our thick and thin filament, the actin and the myosin. So again, the myosin will be in the center, and then the actin will be more on the periphery on either side here. Okay, so you can see here, in a relaxed smooth muscle cell, these structures here are going to be, there's going to be some overlap going on, okay? At the end of each actin, all right, filament, you'll have what's called a dense body. Think of the dense body as like an anchoring point there, okay? So that's what these actin filaments can attach onto, all right? Then we'll also see throughout the intermediate filaments, and they'll also act as an anchoring point because these dense bodies can attach onto those structures. And intermediate filaments another part of the cytoskeleton, all right, when we're dealing with cells. Right? In this case, we're dealing with the smooth muscle cell. So you can see here, if you look at the distance on the inside of our triangular configuration here, all right, now compare it to, on the right side, when that smooth muscle cell undergoes a contraction, we can see an increase in overlapping between the thick and thin filaments, but a decrease here, all right, on the inside of that triangular area there. So the area of that triangle has decreased, all right, on the right side compared to the left side. This is the relaxed configuration. This is the contracted configuration. You can even look at the cell. See the cell, the length of the cell here when it's relaxed compared to the length of the cell here when it's contracted. All right. Yes, it's longer here and shorter here, all right, but it's kind of thickens up. It gets fatter in the center here during that contraction process. Now, a lot of these smooth muscle cells, because of this configuration, are anchored to one another, okay? All right, and these intermediate filaments help with that. All right, so we use these dense bodies that you'll see also, or dense plaques, excuse me, all right, that help to anchor onto adjoining or adjacent cells here. All right, it's one of the many ways that we can actually increase the ability for a contraction. Okay, so keep in mind, we still have our contractile proteins, so that the thick and thin filaments there, all right, but our thin filaments, the actin, are going to be anchored onto these specialized anchoring proteins here. Okay, then we have our dense bodies and our dense plaques. All right, this is going to be, the dense body is going to be within the sarcoplasm. All right, and that's going to be that anchoring point for a lot of the intermediate filaments and also the actin filament. The dense plaques are going to be on the inside of the plasma membrane, the sarcolemma. Okay, and that's what's going to allow other smooth muscle cells, all right, to attach themselves or anchor themselves to its adjoining smooth muscle cell. Okay? All right, so keep in mind, no striations. We do not have sarcomeres. That's a great test question. All right, and if there's no sarcomeres, then there's obviously no Z-discs. All right, Z-disc is part of the sarcomere configuration, so is the M-line, so is the H-zone, the I-band, all that, okay? So keep that in mind. All right. So here's how we're going to see. There's a couple other things I want to mention, then I'll kind of walk you through how smooth muscle contracts. We're not going to go into all that specialized detail that we did for skeletal muscle, all right? But we will see, all right, because there's going to be a cross-bridge formation occurring, all right, we're going to see how actin and myosin are actually going to overlap each other. Now notice, all right, some of the ingredients that we have in our smooth muscle is, all right, you hear me mention actin, myosin, troponin. All right, but wait a minute. There's no tropomyosin. Okay, we're missing tropomyosin, that long, all right, rope-like regulatory protein. But we do have troponin, and that's going to play a role here. We're going to talk about that in a second. All right, so some of the things that smooth muscle has that skeletal muscle doesn't have, all right, is that we're going to see with the myosin heads, all right, we're going to see the myosin heads along the entire length of this baby. All right, which allows for more cross-bridge formation. That's an important concept to understand. All right, so these filaments, all right, that make up that involve the myosin heads have this special, all right, mechanism called the latch bridge mechanism, and the latch bridge mechanism allows for an extended or sustained muscle contraction over an extended period of time. Important, without using extra ATP. That's huge. Okay, that is very huge, especially when you're undergoing certain biological and physiological functions. All right, when we're trying to move food through our small and large intestines through peristalsis. All right, so keep that in mind. This concept here, these filaments, because it has the myosin heads scattered throughout, okay, and it has this latch bridge mechanism, allows for an extended period of time or a sustained contraction to occur without using up all that ATP. All right? So we've seen this protein. This is a calcium storage protein, Calmodulin. We saw that in the terminal cisterna before. Okay? And so this Calmodulin, there was two proteins storage proteins in the terminal cisterna, the calmodulin and the calsequestrin, all right? But here in the smooth muscle, all right, calmodulin is going to be the protein that's going to start off or trigger the contraction. I got a great picture, I'll explain that to you, okay? So this calmodulin, all right, is going to activate an enzyme, all right? MLCK stands for myosin light chain kinase. Well, there's that term kinase again, all right? So we know that kinase likes to stick a phosphate onto something, all right? We call that phosphorylation, all right? So it's going to phosphorylate the myosin head. I'll show you this picture, okay? So this will actually initiate a contraction, okay? So we have to turn off the contraction. Well, not to worry. We've got another enzyme here in the smooth muscle called myosin light chain phosphatase. Another enzyme, but in this case, it's going to strip that phosphate molecule off of the myosin head. And when that does that, it's going to stop the contraction and it's going to cause relaxation. All right, so let me walk you through here. Okay, nice picture here. We'll start off at the beginning. Make it a little bit easier to understand the players here. Okay. All right, so keep in mind, majority of the calcium is outside, all right, of the smooth muscle cell, all right? So an action potential then triggers the opening of voltage-gated calcium ion channels. They're located, here's the calveoli, these guys right here, all right, these little indentations. So in these little channels, or these little indentations, we have the voltage-gated calcium channels. So the action potential comes, hits the voltage-gated calcium channel, triggers the opening of that. All right, calcium then enters into the cell, diffuses across the the gate here into the inside of the cell, and it's going to bind onto calmodulin, which is that calcium protein here. And that calcium is going to cause a configuration state change. It's just going to change the shape of that calmodulin. And what that does is it allows that calmodulin to come on over here, all right, to the inactive, all right, myosin light chain kinase, and it activates that enzyme. All right, it activates that enzyme then. That enzyme is then going to, because it's got kinase in its name, it's going to phosphorylate, basically stick a phosphate onto, all right, the myosin head. And by doing that, all right, that once it does this, it's going to activate the myosin, and we're going to initiate that cross-bridge mechanism. All right, it's a little bit different than skeletal muscle here. All right, so where do we get the phosphate from? ATP. All right, ATP. Okay, this is an energy-requiring process. So we get it from the ATP. The phosphate goes onto the myosin head. All right, the myosin head is now activated. It can now initiate, all right, that cross-bridge formation. All right, and now we can initiate. You know the rest of the story. All right, we get the cross-bridge formation, power stroke. All right, then we'll reset it, and then we'll do it again if need be, or we will relax, all right, depending on what is going on with action potentials and whatnot, okay? So we're going to see, all right, now in this situation, remember, the only way, okay, that we're going to disengage from this cross-bridge formation, all right, is when we get that myosin light chain phosphatase, all right, that other enzyme is going to come in there, and it's going to then cause myosin to disconnect. Otherwise, it's going to stay connected and it's going to continue to contract. All right? So that's why, all right, smooth muscle doesn't need, all right, ATP continuously like skeletal muscle does if it's going to sustain a contraction. All right? It's basically locking something in or unlocking it. And that's pretty much what we're going to deal with there. And it's because of this, this is why we consider smooth muscle to be fatigue resistant. All right? So that whole point, you can maintain that contraction without that ATP. All right? So once you've sustained engaged the contraction, all right, that cross bridge will remain engaged until, all right, that myosin light chain phosphatase comes along and it disconnects that cross bridge. Okay? And again, the neurons that innervate, all right, this type of tissue are going to be involuntary, mainly from the autonomic nervous system. All right, questions for me? Okay. Well, that will be it for muscle. That was a lot. It's a lot of information. All right, you got the whole weekend to kind of take it in, absorb it, swish it around like some Listerine in your mouth, swish it around in your brain with like some Listerine. All right, watch the videos and whatnot.